Hello, my name is Howard, and we are from the Fred Hutch Data Science Lab. Our talk will be on enhancing access to genomics tools. We are funded by the ITCR, a program of the National Cancer Institute and CI. The IT stands for the ITCR Training Network, and its purpose is to catalyze informatics research through training opportunities. Our website is itcrtraining.org. We are all busy as researchers. Technology is changing quickly, and it's hard to keep up. Our guiding principle is: cancer research will advance faster if informatics tools are more accessible. Cancer informatics is hindered by a gap between different types of experts. The IT serves to make informatics easier for cancer research. We aim to improve the preparedness of tool users and also to improve the usability of tools developed by scientific software developers of the ITCR. We make courses about informatics, make software for course creation, provide live education, and enhance community engagement in cancer research. We have courses about management and leadership practices, software development, tools and resources for cancer research, and best practices including reproducibility and ethics. All courses can be found on the itcrtraining.org website. Here is a list of our future courses, including Cancer Imaging Informatics, Cancer Clinical Informatics, Machine Learning for Cancer Informatics, and Cancer Informatics Data Visualization. Concepts discussed in choosing Genomics Tools course. What does your genomic data type represent? What are the most common data processing steps for your data type? Find resources, tools and tutorials to help you process and interpret your data. Here is our Choosing Genomics Tools course. Its goals are to help users identify what genomics tools and resources best meet their data type-specific analysis needs. It is organized to include chapters about fundamental concepts followed by chapters with advice for specific data types. This is a living course like a Wikipedia for genomic data analysis. As technologies and data type handling recommendations change, our course will too. Adding new data types, updating recommendations, adding new tools and methods. We have included the listed data types so far on the slide. We hope to add more as individuals contribute. Thank you for your attention and time. Now we will have a presentation of 2 ITCR funded tools for handling genomics data. Good morning. My name is Susanna Kiwala, and I'm a software developer in the Griffith Lab at the Washington University School of Medicine in St. Louis. Today I would like to introduce our immunotherapy tool suite PVAC tools. A major focus of our work is on understanding how T cells can recognize tumor cells by their unique features. Our goal here is to leverage the immune system's ability to identify and destroy cells infected with non-self or foreign elements. Instead of the foreign element here being a pathogen that has invaded a cell, it is the tumor cell itself. This works because the somatic mutations in tumor cells create unique peptides that can be recognized as foreign. Depicted here is an example of the simplest form of a neoantigen, a single nucleotide change leading to a single amino acid substitution. In normal cells, the wild-type peptide is presented on the cell surface by the major histocompatibility complex, but it will be recognized as self by the immune system. In the tumor cells, if the altered version of the peptide is presented, it can lead to T-cell recognition and ultimately cell death. In personalized neoantigen vaccines, you are identifying a set of these peptides and delivering them to the patient to stimulate an immune response against the unique features of their tumor. Predicting which neoantigens might be presented by a tumor cell and selecting the ones to include in a vaccine is a complex process with many criteria to consider. PVAC tools is designed to be used on top of a well-established variant calling pipeline. It uses a VCF file of somatic variants, annotated with consequences by the ECCB variant effect predictor, to predict neoantigen candidates from missense in frame indoles and frame shifts. Additionally, annotated fusion calls can be used to predict neoantigens from gene fusion events. In humans, the MHC complex is a set of HLA alleles that differ from patient to patient. A patient's HLA type can be determined by in silico tools or via clinical typing. PVAC tools is a set of several complementary command line tools. PVAC-seq is used for predicting neoantigens from somatic alterations. PVAC-fuse is used for predicting neoantigens from gene fusions. PVAC-bind uses a FASTA file of user-generated peptide sequences as an input. The results of each of these tools are 3 main files: a full list of new antigen candidates, annotated with all information obtained during the run; a filtered list of new antigen candidates based on the thresholds set by the user during the run; an aggregated report that selects the best new antigen candidate per variant based on several criteria like peptide MHC binding affinity, transcript support level, transcript biotype, and inclusion of problematic amino acids, among others. Each selected best new antigen candidate is assigned a tier such as pass, low expression, subclonal, etc., based on its characteristics. PVAC-View is our shiny web application. The aggregated report can be visualized in PVAC-View to further review each new antigen candidate and select the ones desired for vaccine manufacturing. Lastly, PVAC vector is used to generate a single DNA vector new antigen sequence by combining the selected new antigens in an order that prevents novel junctional peptides, using spacer sequences if necessary. PVAC Tools is open source, open license and easy to install. It is packaged on PyPI and can be installed with pip install PVAC Tools. We also provide version releases on GitHub and version Docker containers on Docker Hub under the Griffith Lab-PVAC Tools namespace. PVAC Tools is licensed under BSD3 Clause Clear. Documentation is available at pvactools.org. For help, we have an email alias available, or users are welcome to open a GitHub issue. We are also in the process of putting together an IT course on how to use PVAC Tools and have just released the first version. It is available at course.pvactools.org. PVAC Tools is easy to run. It only requires a few arguments: the PAP to your input file, the sample name, the HLA alleles of the sample, the prediction algorithms to use, and the desired output directory. However, it offers a large set of optional arguments to customize your run. The epitope, length, and normal sample name are just two used in this example. The full list is available by running pvacseq run-h. Our IT course has further examples and explanations of arguments we recommend. The results of your PVAC-seq runs can be visualized in PVAC-View. The starting screen has a list of input fields for uploading your result files. It also offers the option of loading demo data for the ECCB1395 breast cancer cell line. The main table in PVAC-View shows the best neoantigen candidate for each variant. It lists the gene and amino acid change of the variant, as well as additional information about the best peptide and the best transcript coding for it. These include, from left to right, the transcript support level, the best binding HLE allele, the mutated positions of the best peptide, any positions in the peptide where the amino acid might be problematic for manufacturing, and the total number of new antigen candidates passing the binding affinity threshold set by the user. If a gene of interest list was uploaded, variants on those genes have their gene highlighted with a green border. Next, this table lists the IC50 peptide MHC binding affinity for both the mutant and the wild type. It also shows the percentile scores of the binding affinity values. For the mutant values, a heatmap coloring is applied to make it easier to visually identify well-binding peptides. The next few columns show the coverage and expression of the best transcript with a bar plot background to represent where specific values fall across the entire patient sample. The tier column represents the tier assigned to the best peptide. The new antigen candidates in this view were all sorted into the pass tier, but tiers such as low expression or subclonal are applied to easily identify why a new antigen candidate might be unsuitable for vaccine selection. The column reference match reflects whether or not the best peptide was found in the reference proteome, which is undesired since such peptides are not novel and including them in a vaccine might lead to an autoimmune response. Users are able to set a status for each candidate in the evaluation column to mark them as accept, reject, or requiring further review. For candidates not sorted into the pass tier, red borders visually highlight the attributes failed by the candidate. Users are able to further investigate each candidate by clicking the Investigate button on the right of each row. For example, they are able to see information about reference matches. This shows details about where in the reference proteome a match was found. Users are able to see details for other peptide candidates resulting from a variant, as well as details for alternate gene transcripts. The best peptide and transcript are highlighted in green in these tables for easy identification. Myelin plots show a per algorithm visualization of the predicted binding affinity of the best peptide to check the congruence between algorithms. These are just a few examples of the detailed visualizations available in PVAC-View. Please check out pvacview.org, our documentation and our IT course to see more. I would like to acknowledge and thank all the members of the lab and the PVAC Tools team in particular, as well as the funding agencies that have made this work possible.
Hello, my name is Alan Lynch, and I'm a graduate student at Harvard Medical School in the Bioinformatics and Integrative Genomics program. Today, I will present work I conducted as a member of the X. Shirley Liu Lab at the Dana-Farber Cancer Institute, where I, along with Cliff Meyer, Christina Theodoris, and Miles Brown, developed MIRA, or Multimodal Integrated Regulatory Analysis. MIRA is a Python package for analysis of single-cell multiomics experiments. Single-cell multiome experiments capture RNA-seq and ATAC-seq data from the same single cells, where each cell's transcriptomic and chromatin accessibility profiles are linked by a shared cell barcode. Leveraging both modalities, these experiments enable unparalleled resolution for investigating the regulatory architecture of cell state and gene regulatory circuitry. MIRA is multifaceted and addresses many of the problems that one might encounter during multiome analysis. First, MIRA includes powerful methods for topic modeling, batch effect correction, and constructing expressively in space representations of cells. Next, MIRA includes methods for pseudotime trajectory inference, with an emphasis on analyzing trajectories and cell state transitions using stream plots, as shown in the second box. Finally, using MIRA's cis-regulatory modeling API, one can investigate the relationship between gene expression and proximal chromatin accessibility, and infer which transcription factors are driving transcriptional states in which cells. In my short presentation, I don't have time to cover all of MIRA's capabilities, but if you're interested, you may read about them in our Nature's Methods paper, linked at the end of my slides. Today, I will focus on our most recent work, where we developed a new method for the representation and analysis of perturbations in multiomics data, which was recently featured by Nature Communications. Combining single-cell multiomics measurements with genetic or drug perturbations of cells is a powerful strategy for investigating cell state dynamics. By observing what changes in response to a multitude of perturbations, one may piece together the regulatory programs underpinning complex cell systems. Typically, an experiment starts with the creation of a wild-type reference atlas, usually in the initial characterization stage of an inquiry. Next, perturbations are performed in one or more subsequent sequencing batches. For each perturbation, the high-level questions to address are what cells were affected, and how did those cells change in response to the perturbation. Unfortunately, because each batch of data is subject to different technical effects introduced by the data collection process, direct comparison of perturbed to wild-type cells is confounded by what is known as the batch effect. For example, we may know that some perturbation affects the highlighted cells, but technical effect confounding makes it difficult to address to what extent. Could this perturbation induce an altered cell state or trajectory? What differentiates the new cells' state from wild type? Past methods for batch integration were insufficient to answer these questions, since most assume that differences between batches are mostly technical, and thus explained away the interesting effect of the perturbation as nuisance variance. We set out to design a more sensitive method that could disentangle the real biological influence of perturbations from differences in technical variation. This culminated in CODAL, or covariate disentangling augmented loss, a new statistical model and novel objective function uniquely well suited for this purpose. In the challenging simulation posed above, for example, CODAL correctly determines the affected cells forge a completely new cell state trajectory. So how does CODAL work? First, we assume that the observations from a single-cell sequencing experiment X, in this case transcript counts for single-cell RNA-seq, are distributed according to log additive factors for true biological variation plus technical effect corruption. We suppose that biological effects are linearly factorizable into discrete cell state programs, or topics, given by the matrix beta. Each topic, or row of beta, describes how genes' expression changes with activation of that program. The latent state of each cell is represented by the Z matrix, which describes which programs are influential or activated in that cell. These topics and cell states are unknown a priori and must be discovered from the data. Next, we assume that technical effects depend on the specific conditions under which the data for those cells were generated. Those specific conditions are almost always unknown, but since cells from the same batch were presumably sequenced under similar conditions, the cell's batch of origin is a useful proxy variable that describes this effect. Technical effects may also vary according to cell state due to differences in cell size, nuclear chemistry, or some other unknown variable. Since the functional form of technical effects is unknown, we parameterize the estimate for technical effects as a neural network H, which depends jointly on cell state and batch covariates. Altogether, CODAL seeks to explicitly separate biological and technical effect influences on the observed data, and further factorize biological variation into interpretable gene expression modules. The key challenge in inferring these effects is shown by the variable dependency diagram on the right. We aim to learn the distribution of expression rates for cells in state Z without confounding technical effects. However, due to the association between expression rates lambda and technical effects T, implied by their dependence on Z, These effects are not independent. Therefore, we propose to approximate the unconfounded distribution of biological quantities by penalizing the dependence between biological and technical effects through regularization of mutual information between their distributions. The result is our novel objective function, which maximizes the marginal likelihood of the data given the parameters of the model while minimizing the mutual information between biological and technical effect distributions. The following test demonstrates the effect of introducing the mutual information regularizer. The plots on the left show the biological and technical effect predictions for the gene CCL5, a CD8 T cell marker, estimated from a bone marrow hematopoiesis dataset composed of 60,000 cells and 13 batches. Using the same statistical model as CODEL, but without mutual information regularization, biological and technical effects are entangled and show an incoherent relationship with the actual observed counts, highlighted in gray. Simply adding the mutual information regularization, or using the CODEL objective, one can see that biological and technical effects are rendered essentially independent. Cell types are stacked according to increasing CCL5 expression, while batches are arrayed according to basal CCL5 counts within them. Importantly, CD8 T cells are shown to have the highest CCL5 expression, as expected. In summary, by regularizing the relationship between biological and technical effects in the generative model, CODEL disentangles signal from mostly independent sources of technical noise. This, in turn, increases CODEL's sensitivity to differences in cell states between batches, allowing our algorithm to correctly detect perturbed cell states while still enabling robust technical effect correction. For an overview of the extensive benchmarking we performed for the CODEL method, please see our manuscript, again linked at the end of my slides. Now I will demonstrate how Mira may be used for perturbed cell type discovery in a real scenario. This data, generated for Pijuan Sala et al., captures mouse embryo development with 180,000 cells. Data was collected in 5 batches, 4 to construct a wild-type reference atlas, and a 5th in which the tau1 transcription factor, required for formation of the erythroid blood lineage, was knocked out. The perturbed batch, shown in blue, exhibits quite strong batch effects that preclude comparisons with wild-type cells using PCA. The right side shows the code to analyze this dataset using Mira. First, I instantiated a model and provided metadata indicating which technical covariates to use. Next, I saved the dataset to an on-disk cache to reduce memory overhead. Finally, as with any topic modeling-based application, one must choose the number of topics to represent the data. Here, I've used a gradient-based estimator, which automatically selects the number of topics using only one training pass through the dataset. I then fit the model using the selected number of topics and continued to the analysis. As you can see, Mira effectively removed batch effects from the latent space representation of cell states. The perturbed tau1 knockout batch was successfully integrated into the wild-type atlas for comprehensive downstream analysis and comparison. The integrated latent space preserves the exploding complexity of the developing embryo and discriminates between the cell types annotated by the author of the original study. So how did the cells in the perturbed embryos change? The perturbed embryos were actually chimeras. Since completely knocking out tau1 would have been lethal, the authors fused gene-edited tau1 knockout embryonic stem cells to a wild-type blastocyst. The embryo subsequently developed with some fraction of cells which were perturbed and some fraction of cells which were wild-type. In the UMAP on the right, one can see chimeric wild-type cells develop normally, while chimeric delta tau1 cells failed to contribute to the erythroid blood lineage as expected. Interestingly, while most other cell types appeared unaffected by the knockout, the hematoendothelial progenitor group showed many differences in cell states between wild-type and delta tau1 cells. Zooming in for a closer look, we identified 5 distinct subclusters where entirely new cell states emerged as a result of the knockout, only 3 of which had been found in the previous study using a more ad hoc procedure. Introspection of each subcluster using topic analysis revealed that the new cell states were in fact hematoendothelial progenitors, which had reactivated other gene expression programs found throughout the mesoderm. To conclude, the Mira package for single-cell multiomic analysis and regulatory inference is now equipped with the CODEL algorithm for integration and characterization of perturbation experiments. We anticipate that this will be a useful tool for dissecting complex regulatory circuitry.